one I have seen here from Sundarasan, what type of sequencing method is adopted and whether de novo assembly was executed to compare the transcriptome. The thing is this, the 2012 paper, when the first genome sequencing was done, that was de novo sequencing, and now we don't need to do de novo because already a gold standard genome sequence is already available. So we do resequencing with a very cheaper price, so that you can just map the reads on the already sequenced information. The next question is how osmotic effect of salinity reduce plant water uptake. This is physiological question. Next one is there any hidden medicinal potential in foxtail millet? This needs to be addressed because there is no publication, at least with justification about the medicinal potency, but I hope it should be, because nutritionally and in other traits it is highly efficient. It also should be looked out. Is there any effect on human being on transgenic millet? There is no information on transgenic millet because we are developing for the first time, and it should be tested in different RCGM protocol of Department of Biotechnology. So until unless we test, we cannot say. But in general, if I say from other crops, till date there is no negative effect has been reported for any transgenic crops. So the next question, quality parameters and storage potential of millet. Is there any genes for seeds longevity are identified in millet? No, this information is very important, but we are also working on it for seed longevity. Yes, this is very important. So, Sanju from, I don't know from where, Surjit Sanju, he asked that whether foxtail millet can be used in diabetic patient. Yes, that is the characteristics of all the millets, that their glycemic index is very low and they contain good resistant starch. So the glycemic index becomes very low. So what you do when you eat the carbohydrate, and due to presence of good resistant starch in all the millet crops, the conversion of carbohydrate to glucose is slow. So the transcription of glucose in the blood is become slow. So all the millets, due to presence of good resistant starch, they can be a good use for the type 2 diabetic people. Can foxtail millet be a substitute for rice consumed on daily basis? Yes. If you go in the southern part, now you will get ragi dosa and everything, because nutritionally this crop is 5 to 6 times better than rice and wheat. And then in top of this, there's no need of taking proper care of these crops, because they grow in marginal land where there is no need of fertilizer, no need of water. So nutritionally better 5 to 6 times. Very less water is required. So I think millet is the substitute for all the major crops. So today's lecture I have given to show that how important the millets are as compared to the rice and wheat. Which is the better consumed for oats and millet? They are all same, but millet will be better. Joseph, so my question is, when it comes to identification and overexpression of genes, there are many genes involved. Why did you focus on working genes? That is the reason I told you that the story of hits of protein. 113 hits of factors genes have been identified. We cannot characterize 113. So that is the reason we developed this pipeline, bioinformatics, through HMM profile and bioinformatics tool, to identify the different classes. Like you asked me for the working genes. For working genes, we have identified more than 100, but we cannot characterize more than 100. So we test these genes for different stress, and then we find out the potential candidate which has been showing differentially expression in most of the stress. So like in HSP, I have showed this HSP27 showed differential expression in all the 4 states, like heat, drought, salinity, and so and cold. So this is the potential candidate. How we can pinpoint the number 1 or 2 or 3 genes. Sir, is there any study on reported of qualitative traits in foxtail millet? No, we are doing all quantitative traits, but I think there should be, because if I think TNU, some Ravi Keshavan and Bandi Rajan, they are doing the qualitative traits in foxtail millet, but not only foxtail millet, they also work on podo millet and barnyard millet. Anti-nutrients such as phytic acid and tannin present in this tuna. This is a very good question by Prabhat Kumar Singh. Anti-nutrients such as phytic acid and tannin present in this millet, can we improve this using genomic? Yes, and any improvement in this area is going on. We are working on it, and my student Muthu has an ECRA project on phytic acid in foxtail millet, and how to reduce this phytic acid content in foxtail millet. He's working on it, and we identified one candidate gene, and we try to actually silence this gene so that the level of phytic acid we can control in foxtail millet, because this is the problem for rancidity. So if you keep this millets for longer duration, it tastes bitter. So how do select favorably regarding the trait of interest in GWAS? Because that is the reason GWAS will give you an overall idea, but with this, when you try to find out the candidate gene for the most tagged one, most linked one, then we have to get an idea of what are the candidate genes that has been identified through genomic approach through GWAS. What are the necessary steps taken to commercialize the popular of foxtail millet among people in the world? So you can see that if you go to the market, in supermarket, you will get all the millets, but their price is more than rice and wheat, because the production is less, and the people for other countries, but mostly the European and American, they are liking millets due to this nutritional property. So that's why in India, the cultivation is less, production is less. That's why the whatever production we have, the price is very high, because demand is there. So as I showed you one story about this Bandi Rajan, Professor Bandi Rajan from TNU Madurai campus, he developed a hybrid barnyard millet that has replaced the rice and that in that region. So by this way, we can commercialize and popular foxtail millet by showing good improved quality of millets in both in terms of production and quantity. Sir, can the toxin in millet can be reduced by identifying those genes in manipulating them? If so, how? So as I said, the toxins in millet like tannins and then the phytic acid. Similarly, we have to phenotype the different toxins in the range of germplasm, diverse germplasm, and then we have to do genotyping like GWAS. And then we have to identify the genes, candidate genes, which are linked with these toxins. And then we can identify the candidate gene based on the GWAS study. How can we such millet get available in Nigeria soon? I think ICRI said that the CGIR system, they are using a lot of money, investing a lot of money for this millet crop grown in all this sub-Saharan Africa. So I think this millet is now available in Nigeria and other African countries. They are well grown.